Good morning, everybody. Today we will start with another tutorial, MQSS and Perseus tutorial, focus on isobaric labeling. We already talked about isobaric labeling. We talked about that last Tuesday. The theory part, Hamid presented to you an amazing, lecture about that. And today we will make in practical exactly what he presented. Most important, when he was finishing his lecture, he was talking about the MQSS weighting normalization. And he was explaining that there is these features in MQSS that you can use. That you can use it when you have in your isobaric labeling dataset what they are called reference channel. However, we will see here, but also in the paper that is referred to this weighted normalization, it was developed for that purpose, but you can also use it when you have in your dataset reference channel. When you don't have, you have to put all the flags that you have as a reference channel, but we will see together. I will open MQSS and I will show you that one. However, today I will use a dataset. We will see the experimental design. So we will start exactly from a paper, run that one in MQSS, and then do the downstream analysis in Perseus. But we will see first the effect of this MQSS weighting normalization. For that reason, you can see already in these slides that I will show you how to run without the weighting normalization. So just the basic isobaric labeling, you will say to MQSS the quantification, and that's it. But then we will see also the option when you apply the weighting normalization and you define the reference channel as they are stated in the dataset. So let's say if the first channel is the reference channel, you will select the first one, but we will also see the opposite. So I said that this example where the first channel is the reference channel, we will see also the opposite. That it means that you will take, let's say that is 6 plex, you will take from the second one to the last one. So all the others except the first one. And I will show you later the reason of it. Let's first start from the data. So as I said, we take exactly proper data. So something that it was published, it was not done by us. It's a lab located in United States. And this is the paper, a quantitative proteome map of the human body. You can already see in this picture that it's an isobaric labeling, TMT MS3, 10 plex, where the first and the last channel are the reference channel. And plus, you can see already from here, from this other picture, picture A, that it's exactly as the title says, it's a proteome map of human body. So we have several tissues that are from the human body. But let's stay more specific. So if you will read this paper and you will check also the supplementary material, you will find exactly this one. So the experimental design of this. And this is exactly from the paper. I didn't change anything. I just put it as slides to show to you. And you can already see. So there are different runs. Here there are, I show to you just for the resolution, the first one and the second one, but there are, you see, from 1 to 56 runs. There are the TMT tags. As I said, it's 10 plex, so we have these 10 tags. And we can see that for each TMT channel, it says to us what is that one. So we have the reference. So like as I said before, it's the first one, the last one, the one that here I highlighted in yellow. But you can see that already comparing this first run and the second run, that in the other channel I have all the other tissue, but they are not distributed like that. In the first run, I have only the stomach. So all the channels are referred to stomach, and they run through only to esophagus, let's say, but you see that it's mixed, okay? And at the same time, so in each run, I can find several tissue, and at the same time, exactly in this example, if I take, for example, the run 1, I have in the second channel muscle, but as soon as I go to the second run, I don't have muscle in the same channel in the second one. I have that one in the fourth one and in the 91. Okay, so it's mixed. It's heterogeneous because there are many tissues of the human body, but at the same time also exactly these tissues are mixed in our runs and also in our TMT tags. Okay, we have this experimental design. Okay, we have this paper, we have these studies, and we have this. Let's go to MAX1. We want to see this free possibility process. Okay, so this one is one process, the one in the middle is another one, and the third one is another one. Just to repeat, the first one is without normalization, so just standard isobaric labeling. This one is normalization, taking this to reference. And then the one that here we call sample is the opposite. So the sample where we have all this tissue. Go to MAX1. And you have the data, so you can do that together with me. Okay, so let's open MAX1. We provide in the USB stick, so the link that we sent you via email. And as usual, MAX1 you can find under software. And we just open MAX1. Okay, we have MAX1. First, we have to load our raw data. I provide to you the raw data, and there are just press here, load. And you go to the USB or where you located the material that you download. In my case, it's here, data. And I have this one, practical 8 isobaric labeling with MAX1 weight and normalization. You open this one under data, and you will find these 4 raw files. So just select them and open. So these are my samples. Of course, if you check with the paper, you will find that you will see that there are not only 4 raw data. This is just an example. There are 100. I don't remember exactly how many, but this is exactly an example for you to see how to do that. So as you can see, I have 2 fractions for 2 instruments. You see, for 2 kind of sample. So what I will do is that for now, we will do the one that is without normalization. So just an isobaric. So I have to set my experiment, and I have to set my experiment is the one that it's called in the slides that it was called the run sample. And what I do is that I select the first 2, set experiments, and I can call it S01. Let's say just very basic. You can call it as you want. Just you need. Yes, sorry. Yes.
Unless we.
You have 2 files. You have the first one. Okay. Okay, just select only one. Probably it was because of the size of the USB. Sorry.
Maybe can you remove also the other 2?
Yes, maybe I can remove. Exactly, good idea. So I guess you have this, correct? So in this case, you click the first one, set experiments, and let's give just S01, or it's better that you call it as it's called in the experimental design, so that then you remember to which one is referred. But for now, it's just an example. I just put it as 01. But the second one, because it's a different one, I will put a different name as 02. Okay, we have our data. We define the center of experiment. So we don't want to use the weight normalization for now. We want just to quantify as isobaric labeling. So here it's done. We don't have to do anything. But we need to tell to MAX1 to which quantification we have. We saw before that is a TMT MS3. So we select a reporter under group specific parameters, reporter ion MS3. It's templex. So I press here, templex. This is important. So here you see that there are the correction factor. Be careful all the time to check if there are possible correction factor in your in the data that you use. In this case, we don't have, but just before to do that, just check this one if you have to change it. So we say to max on which quantification we have. Fine. What we have to do, as usual, what also Hamid showed to you on Tuesday, we have to give the FASTA file. The FASTA file is Homo sapiens, so we have to give Homo sapiens FASTA file. So we go add global parameters, add. And you have them under FASTA. I guess you have FASTA, I guess. And you just open them. That's it. And doing this, everything is done. Just be careful here. I don't know if I'm correct, but that one, number of threads. So here you have to check the size of the memory that you have in your server. Now, I will not start to process, but I would like to highlight one thing. So here you see number of threads, and here is selected as default 4. You can change it, but it depends on which server laptop you use. You have to check the memory that you have, okay? Each thread takes 2 GB when the quantification is DDA, so the DDA LFQ, DDA TMT, Isobaric labeling, except DIA. When you process DIA, it's for 4 GB for each, okay? So, and just as basic example, if you have a server that is 200 GB of memory, you will put no more than 100 here, okay, if you process this one. But if you process a DIA, it's 50. You have to select 50 or less, but no more than 50. Here it's just example, we not change it, but just be sure about this one because it's extremely important when you process a data set in Maxwell. Then we did so we configure the Isobaric labeling data without the without the weighted normalization. Let's do that with the weighted normalization. Let's do the other case when we can say when we put as reference the reference that we have in our data set. So this remains the same. So we say to Maxwell the quantification and everything. What we have to do is 2 things. First, set this reference channel. The reference channel in our data set was the first one and the last one. So the 1 and 10, the first one and the 10th one. Very basic. It's very basic how to set this. So you just select all your raw files. Here you have 2, so just these 2. You go here, set of experiments, and you add the channels that are set of experiments. That are, sorry, reference channel. And in my case, it's 1 and 10. So we'll do 1, add, and 10, add. I define the reference channel. Now I go under group specific parameters, and I go here in normalization. And I have to say to Maxwell that he has to run this with the weighted normalization. And here, instead of none, I select weighted ratio to reference channel. So now we configure Maxwell with the Maxwell weighted normalization with the reference channel that we have. I'll show you that we have a third case. So taking as a reference channel the samples, so the ones that are not reference channel. So what you have to do is that here is as usual. So this one remains like this, but here we have to remove this and put the others. So what we do is that we select these 2. We just first clear. And if we take the opposite, it means that we have to add 2, 3, 4, 5, 6, 7, 8, 9. So one by one, 7. Done. And this is the third case, how to do that when we take as a reference channel the samples, okay? And we will press start and process this one. This, as I said, is 3 different Max1 processes, so it will give you 3 different TXT output, okay? Today, during this tutorial, because we don't have enough time, we will take, I'll provide to you and we will use that one of the output. That is exactly this one, when we take as a reference channel the samples. So not the reference channel as stated, but the opposite. And so what we do is that this one, we can close it. We go to software and we open Perseus, because now we will do the downstream analysis of the output of this quantification. We open Perseus, as usual, as you see also yesterday. You upload here and press select. I provide to you the output, and you already see here in the directory. It's under practical 8 isobaric labeling with Max1 weight normalization. Output, not data this time. Protein groups. Yes, there is a Pelly, there is a one question.
Thanks. My question is about the step just before. So you run first the raw data with the reference channels 1 and 10, and then once that is run, you run the rest? Or?
Yes, so you do that in 3 different Max1.
Ah, okay.
Free total. Now, I change it in the same one because to show to you the configuration, but are 3 and 3. So you open 3 times Max1. One is without the weight normalization, one is with weight normalization with reference, and this other one with as reference channel the samples.
Okay, thank you.
But it's free. Then if you want to process in parallel, depends on the RAM, it depends on CPU that you have in your server, but there are 3 totally different ones. So I open Perseus. Here, as usual, upload this red arrow. Select, and it's this one, practical 8 isobaric labeling with weight normalization. Instead of open data, we open output, and we open protein groups. Now, our TMT channels are called in the protein groups file reporter intensity, sorry. And you can already see that there are the corrected and the one that where it's not written, just the standard one where it's not written corrected. Which is the difference? I showed to you that you can change the correction factor if your data set needs. If you do that, you have to take the this one, the reporter intensity corrected. Otherwise, you take the one without. In this case that we don't change, you can double check if you want later. You will see that this the default, let's say, reporter intensity are exactly the same as corrected because you didn't apply any correction factor. So if you take this one or this one, for this kind of data set, it will be the same. But properly, it should be that you take the one where it's not written corrected. We select all of these where it's not written corrected, so all our channels that we have. Here you see more of them compared to Max1 because, as I showed you before, there are more set of experiments. For you, I just provide just only 2 raw files and I put just 2 set, but you saw before in the slides that there are 58 sets. I select all of them. There it is. And here, with main, I include them in the right panel. When I've done, I press just OK. So now we will do a step that you also did it together with Carlo yesterday. We will sort of clean our data. We will remove the all identified by sites, the reverse potential contaminant. If you remember from yesterday, to do this, you select the matrix called the matrix 1, filter rows based on categorical column. And you just press here. We have to remove all identified by sites, so let's remove it, and it will reduce the matrix. We do that once again, this time for reverse. And then potential contaminants. Okay, now we saw in the slides that we have in each channel a different tissue, and actually in some of them we have this reference channel. If you see this matrix, Perseus doesn't know that, so it doesn't know that in this report intensity 5 there is the tissue reference. Okay, we need to tell him this. We can do that with the annotation rows. And how you can do that is that we also did it yesterday, just repeat. Annotation rows, categorical annotation rows. So here there are several options. What you can do is that, you change here, but you have to change for 560 column. So if you want to do that, if you have time, do that like this. But we have also another solution, much faster, and it's the way to create a template. But first, we need to write this template in a way to say that in each reporting test, in each column, there is this. So we need to write it. We will write separately in Excel, for example. And then we'll give back this one to Perseus using read from my file. So for now, what I will do is write template file, okay, and I select where I want to put it. Yes, here you can give the name that you want. For now, I'll just leave as groups and save. Okay. We want to change this, so we want to give all these details to Perseus. As I said, the best from my point of view is that use Excel. And open the file that we generate. And in my case, I put it under here. Output, not Excel file, all files, this one. So here you see already that there are two columns. Name is the name of your columns, and that one you don't have to touch it. Otherwise, when Perseus will match, it will not recognize, it will not add the annotation to that channel. So this one remains like this. In new grouping, you will add all this information. But what is good about this one is that you can do new grouping, this one, but you can add more at the same time in the same one. An example, it's one that I already provide to you, that if you open, we can open in Excel, is the one that in the output folder is called annotation rows. And you can see that here there are all the groups. So here there is the name of the columns, and I didn't touch it because I don't have to touch them. And then I add all the others. And you see already that, for example, this first one, this second one is exactly the one that I show you in the slides. So with Excel, you can do this one for as you prefer. You can add how many annotation as you want. For us, for today, it's already prepared for you. So first, let's close it. because you have to close it, because otherwise Perseus will not open. So because it's open with another program, so you have to save and close it. So we have our notation. We go back to here, select our matrix, annotation rows, once again, categorical annotation rows. And this time, instead to write, we will read. So I press read from file, select, and we take this annotation rows. And there it is. It's exactly the one that we saw before in the Excel. Very fast, and you can treat it as you prefer in Excel. Done. So we clean our data. We add our notation. So let's start to do more proper essay analysis. First, let's transform our data. And as you see, as you saw yesterday, to transform our data, we can just go under basic, transform, and we will apply the logarithm to our channels. Then, let me just move here on the top. Now, we don't need anymore the reference channel. So the reference channel, max on need them. When you run this weight normalization, now we don't need it anymore because it's a mix of all the tissue. We don't recognize them in this channel. So we need to remove them. Thanks to the annotation, we don't have to select each column that we have to remove. But thanks to this annotation, we can remove it very easily. So filter columns. The first one, filter columns based on categorical row. My reference are under tissue name, the annotation that is called tissue name. And I will take the one that are called the reference. I will put here so that it will Perseus will remove this one. For this data set, we have also some empty channels, so what they called blank. Sometimes it happens that they do like this. So for this reason, let's also remove this one because we don't need them. It's empty. There is not a reference channel. There is no tissue. We don't need. And so we use this one. It will remove this one. It will reduce the matrix, so we don't have to change anything. So just press OK. Nice. Now we have to do what we did yesterday, filter based on valid values. How we do that, as Carlos showed you yesterday, we will do that filter rows based on valid values. We will do that once again based on the percentage. And for now, for this one, it's okay, the one that is already in default. So we will take 70%. And yes, and it will reduce the matrix. So we don't change anything here. Good. So we clean out, we remove the all identified by sites. We add our categorical notation. We transformed. We remove the reference channel. We filter. Now we want to use a dimensionality reduction. That in our case it's UMAP. The one that yesterday during the Perseus plugin tutorial I showed to you in a way to install a Perseus R because we need the Perseus R to do that one. However, you can already see from here there are some missing values. And for that reason, we need to remove these missing values. For that reason, we need to impute our data. And how we do that? It's under imputation, replace missing values from normal distribution. We don't have to change anything, just press OK. So we imputed our data. And as I said, we want to do this dimensionality reduction, UMAP. As we saw yesterday, we have this one under clustering. So just be sure that you select the matrix. We go under clustering, UMAP analysis. We saw yesterday that here we have to give an executable that sometimes it depends on the laptop Perseus will be recognized automatically. Otherwise, we need to give the directory where our R is installed and where our Perseus R is installed. Okay, that we did yesterday together, and this is extremely important to do this. As you see, also this PC doesn't recognize where R is located, but we can tell to Perseus where it is. I saved, just to do that a little bit faster for my case, where I have that one on the programs, yeah, R, the same passage that we did yesterday. So R, the R version, bin, R script, and it's green because we said we have R and we installed the Perseus R. We saw it yesterday. This done, great. We have Perseus R. Now we can do our UMAP analysis, and so we just press OK. Now we wanted to see our UMAP visualization. We can use scatter plot. So just press scatter plot and there it is. You can see a very nice, let me say, UMAP visualization. And what we can check is that here under categories, we have all our annotation that we had before. So we can go, for example, in tissue name. I check, for example, where is colon, this one, then this one. And we can see that it's clustering very good. You see heart and everything. Yes. What I would like to tell you is that as I show you, as I told you, I showed you before, we did these 3 cases in Maxwell, and now we did this one with the one that was with the samples. So not the reference as stated, so the first one, the last one, but the samples, the opposite. Why? Because you can already see this one. So this one, basically, it's exactly for each of these UMAP, I did exactly the same downstream analysis that we did together now. And this one, it's exactly the one that I show you in Perseus. Here is colored based on the tissue. And here is colored based on the set of experiments. So the one that we called the S01 before, S02, and so on, that we have almost, yes, we have 28. And for sure, maybe I can do a zoom. You see that it's not clustering based on the sample, the set of experiments, sorry, but for sure based on the tissue. But this is exactly the case that we did, okay, together. But here you can see also the other case, when I took the Maxwell normalization with reference, and also when I didn't use the normalization. You see that there is a huge difference of that one. For that reason, we suggest to you to first test this one and yes, try to see if you can get the same result as we see here. Then, yes, for the rest, I think I don't have time, but yes, I gave to you, as I said, the output of this one. But you can do also this one. The raw files, please download from Pride, because I didn't provide to you that one because it was huge. But you will see that if you do that, you will get the same results. And yes, maybe add zoom of the last one. You can see that it's pretty good. Thank you.